We will finish our discussion of Chapter 14 by covering the topic of meiosis. In this part of the chapter, we will begin with an overview of meiosis, which is really a reduction in the chromosome number. We'll talk about a key event that happens during meiosis, which is synapsis and crossing over that takes place. And then I will walk you through the various phases of meiosis, and this includes meiosis 1 and meiosis 2. As we begin our discussion of meiosis, it's important to keep in mind the cells that we're talking about. When we look at meiosis, we're specifically looking at either egg or sperm. And one thing that's important to keep track of are the number of each type of chromosome. So in the previous chapter, or previous part of our chapter, when we talked about mitosis, we were looking at somatic cells. So these are all the cells in your body, with the exception of egg and sperm. And so when we had a cell enter into the cell cycle, it had 2 of each type of chromosome, one coming from mom and one coming from dad. And by the time the cell exited the cell cycle, we had 2 identical daughter cells relative to the parental cell that entered into the cell cycle. That will not be true for meiosis. In meiosis, we have a cell that's going to enter into the cell cycle, but by the time we're done, we are going to have half the number of total chromosomes in the cell. So by the end of this process, each sperm or egg will be haploid as opposed to diploid. It's only going to have one of each type of chromosome. And again, this makes sense if you think about it, because when the egg and sperm come together, if they both started with 2N, by the time they came together, you would have 4N, which would be very problematic. So in the process of developing mature egg and mature sperm, we're actually going to halve the number of our chromosomes so that we end up with a fully mature sperm or egg that has 1N, such that when the egg and sperm come together and we have the fertilized egg, it's now 2N. So again, the result of meiosis is we're going to go from a cell that has 2N to a cell that has 1N. All right, so really the big picture, if you can keep this straight, is that for mitosis, we're talking about the somatic cells. When it goes through this process, the cell entering the cell cycle starts with 2N, and what we end up with is a cell exiting as 2N, but not just one cell, we're going to end up with 2 identical daughter cells to the initial parental cell that entered into the cell cycle. On the other hand, when we're talking about egg and sperm, a single cell that enters into the cell cycle, it starts out as 2N, but by the time it exits the cell cycle, we will actually have generated 4 cells, but they will each only have one complete set of the chromosomes. So what we see is that when we have an immature sperm or egg, such as an oocyte, these will start off as 2N, and then during the process of meiosis, we will generate either 4 eggs or 4 sperm that each will be 1N. With oocytes and this whole process of oogenesis, where we're generating the egg, it's a little bit more complicated. We actually only have one fully formed ovum. We also will generate polar bodies, and these will ultimately be disintegrated. One really interesting thing about the development of eggs in female is that a female is born with all of her eggs, and it isn't until puberty that the eggs will actually progress through the process of meiosis. So each cycle, a woman will have 1 egg that then will complete this process of meiosis. Okay, so big picture, before we get into the specifics of meiosis, if you can keep this slide in mind, this will help you tremendously. So we have a very simplified cell here, just showing one particular pair of chromosomes, one coming from mom, the one depicted in red, one coming from dad, the one depicted in blue. And so again, this would be much more complicated. We'd have 23 different pairs of chromosomes and the X and Y chromosomes, but here we're just looking at one. So let's just say this is chromosome 3, just for the sake of argument. So once a cell is going to enter into the cell cycle, it's going to go through G1, S, and G2, just like we talked about the cell cycle previously. And you know that after the synthesis phase, we are going to replicate all the DNA in the chromosomes. So it's going to look more like this. So this is a cell that has now gone all the way through G2 and now is going to enter into meiosis. And what we see is that we go through 2 rounds of meiosis. The first we call meiosis 1 and the second we will call meiosis 2. In meiosis 1, what happens is we have separation of homologous chromosomes. So chromosome, whatever we call this, chromosome 3 from mom and chromosome 3 from dad are going to separate. And then in meiosis 2, what we have is the separation of the sister chromatids. So this is a little bit more similar to what we talked about with regards to mitosis. So again, meiosis 1, we are separating homologous chromosomes. Meiosis 2, we are separating sister chromatids, such that by the end, any one of these 4 cells does not look like the initial parental cell that we had, which had 2 copies of chromosome 3. Now we've got 4 cells that we generated, each with only 1 copy of chromosome 3. So if you can grasp this slide, it's extremely important and will really guide you through the rest of our examination of meiosis. All right, so again, in meiosis 1, what we're doing is separating homologous chromosomes. So really we're going from a diploid cell to a haploid cell in that first cell division. In meiosis 2, on the other hand, what we're doing is separating sister chromatids. So we are not splitting the number of chromosomes that we have. However, we are having the amount of DNA in that cell. So again, we are going to end up generating 4 haploid cells that each have 1 copy of every chromosome in the cell. All right, so that is an overview of meiosis. Now let's talk about one of the really unique components of meiosis, which is what happens during crossing over in meiosis 1. So in meiosis 1, all the chromosomes are going to come together, and the homologous chromosomes are going to come together, and they can form something called a bivalent. And when that happens, you can have crossing over that takes place. So again, if we're calling red the chromosome from mom and blue the chromosome from dad, the chromosomes can cross over at these regions of homology, and such that what you end up with are very unique chromosomes that are not identical to what went into this initial process. So I've written that out here in a little bit more detail. So if this is chromosome 1 from mom, and let's say this is chromosome 1 from dad, after S phase, it's going to look like this. We're going to have replicated all the DNA. And so here I'm looking at 2 different genes that happen to be on this chromosome. And I've given them different forms of the allele on mom versus dad, just so that we can keep track of what's happening with these genes. So again, at regions of homology, we're going to have a crossing over event that can take place. And such that what we generate here is, for this chromosome, we have big A from mom, it crosses over, and then it's going to pick up this portion of the chromosome from dad. And so we call this our recombinant, and this still looks like that initial chromosome from mom. So this would be looking like mom, the parental strand. This would be a recombinant. Here we had a crossing over event going from the little a from dad to cross over and pick up big B from mom. And then here we have another parental strain that looks exactly like what dad had initially. So again, you can end up with 4 different haploid cells that all are going to look different from one another. All right, so now let's talk through the actual phases of meiosis. So each phase of meiosis is just like in mitosis, where we had prophase, prometaphase, metaphase, anaphase, telophase. We're going to have that here as well, except unique things are happening in meiosis 1 and compared to meiosis 2. The key thing to remember again is that in meiosis 1 we're separating homologous chromosomes, and in meiosis 2 we are separating sister chromatids. So some of the unique things that are going to be happening in meiosis 1 are highlighted here as well. So in prophase 1, that's where the homologous chromosomes are going to come together, and this is where the crossing over event can take place that I just explained. In prometaphase, what we're going to see is that the nuclear envelope is going to begin to disappear. The bivalents are going to attach to the kinetochore, and we're going to start to line up those chromosomes at the metaphase plate as they kind of pull back and forth via those microtubules. The metaphase plate is going to be formed, and what we see here is that all the homologous pairs of chromosomes are lined up at that metaphase plate. And this is going to be very random. I'll explain what that means in a minute. Then we're going to enter into the anaphase, which is where those homologous pairs are going to separate, and then we're going to have the reformation of the nuclear envelope as well as the separation of those two cells, the cytokinesis. So two of the places that we are able to really increase our biological diversity is through prophase 1 as well as metaphase 1. All right, so here again, this is prophase 1. This is when that crossing over event is going to take place. Those homologous chromosomes will come together. Say chromosome 1 from mom and chromosome 1 from dad will come together. Meanwhile, chromosome 8 from mom and chromosome 8 from dad will come together. And we're going to have those crossing over events for all the chromosomes that will simultaneously be happening. In prometaphase, again, we're going to have breakdown of the nuclear envelope. The formation of the kinetochore and the microtubules are going to start to pull back and forth those chromosomes until they line up at the metaphase plate. Now, we say that random alignment takes place. So what you see here is that on this left-hand side of the metaphase plate, we've got one chromosome from mom, mostly from mom, and two chromosomes from dad, whereas on the right we've got one chromosome from dad and two chromosomes from mom. So how chromosome 1 lines up at the metaphase plate has no influence on how chromosome 2 lines up at the metaphase plate, or how chromosome 3 lines up at the metaphase plate. So this random alignment is going to also increase our genetic diversity. In anaphase 1, again, what we are doing is pulling apart those homologous chromosomes, and then in telophase and cytokinesis, we have the formation of the nuclear envelope, and the cleavage furrow will form, and we will pinch those two cells and separate them. Now, in meiosis 2, we have a couple of key steps that are taking place here as well. Again, we've got those same steps, prophase, prometaphase, metaphase, anaphase, and telophase, but we've already separated our homologous chromosomes. So what's going to happen here is, again, the separation of sister chromatids. So this is going to look very similar to what we did in mitosis. So in prophase 2, sister chromatids are going to begin to condense. We're going to start to form those spindle fibers. Prometaphase 2, the nuclear envelope is going to disappear. All those chromosomes are going to now line up at the metaphase plate. And again, we're going to have random orientation of the sister chromatids, which is another way that we increase our genetic variability. In anaphase, sister chromatids are going to get pulled apart, and then in telophase cytokinesis, again, nuclear envelope will reform, and then we will form the cleavage furrow again and generate 4 haploid cells. So as I said, this is going to be very similar to what we saw in mitosis, where we were separating sister chromatids. The key difference here is that in mitosis, we're going to be separating 46 pairs of sister chromatids, whereas in meiosis 2, you're separating 23 pairs of sister chromatids, because in meiosis 1, we already separated homologous chromosomes. So here is what it looks like during prophase 2. So remember, at the end of meiosis 1, we have already generated 2 cells that are each haploid. So now what we have is 2 cells entering into prophase 2. during prometaphase, we're going to have the loss of that nuclear envelope, and the kinetochore is going to get attached here to those microtubules. We're going to pull back and forth until we line up those chromosomes at the metaphase plate. And then those sister chromatids are going to get pulled apart during anaphase, and then ultimately we're going to reform our nuclear envelope and cytokinesis will be complete. So if you notice here, we have 4 different haploid cells, just one copy of each chromosome, and no 4 cells are identical to one another. So this is really how what makes you you is so unique. It would be impossible to regenerate who you are, which is really incredible. All right, so again, if you look at the initial cell that went into this process, we had 3 different chromosomes, 1 set coming from mom and 1 from dad. So we had 6 total chromosomes in this cell. And what we end up with are 4 haploid chromosomes, so 3 chromosomes again, but only 1 copy of each chromosome in the cell, and no 4 cells are identical. All right, so just in summary, I want to make sure that you understand some of the ways that we have our genetic variation. The key is that crossing over takes place in prophase 1 of meiosis 1. That's when those homologous chromosomes come together, and then we get the crossing over event. Independent assortment of chromosomes 1 during metaphase 1 increases our genetic variation, as well as random fertilization of 2 gametes. What egg and sperm come together is completely random.